are biological polymers. So if we start with a related compound, which everybody else has obviously heard of, cellulose, which is a polymer of glucose. We have a ring structure. And this just sort of continues along. This is just part of what goes on in an overall chain. And I'm going to focus on this part right here. So I'm going to write that in a different color. Let me do it in red so it's visible. So this material right here, or this structure I've drawn, is glucose, which forms the polymer cellulose, which is the most abundant biological polymer on the planet. If we look at chitin, very, very similar to this. So we start off with somewhere in the middle of the chain. It's just a long chain. It's the same six-membered structure. That's a 2. And on that particular carbon atom, all these little joints here are carbon atoms, we have this structure. Oops, I messed up already. Let me do something else here real quick. Do a different color. There's a nitrogen in there. And we have, let me go and use another color. All right, so that's the basic repeating unit in chitin. And this base substance here has a quite long name to it. It's N-acetyl glucosamine, UCOS. Now when we get to chitosan, we start off with this structure. When we go to chitosan, what we're doing is we're going to remove this portion, which is called an acetyl group. And the process from chitin to chitosan is a process called make sure I spell this right. Deacetylation. So we're removing this acetyl group. So if we look at that base structure, the material is now glucosamine, and that's related to chitosan. And down here, let me get my right color, we have that. So this is the basic chemical difference between the structures of cellulose, chitin, and chitosan. The term pH, properly written small p, big H, is a measure of acidity in a solution, a substance, or whatever. We're all familiar with this term pH. We hear about pH balance in shampoos or whatever. So what do we mean by acidity? Acidity is a feature that's brought about by the concentration of what's called hydrogen ions in a solution. Acids give hydrogen ions in solution. So that's what this is all about. So the pH number that we all associate with various things is related to the concentration, and to get nice and chemical here, we put square brackets around this thing to indicate concentration. pH is measured along a scale from 14 to 0, and we have a midpoint of 7. So anything pH range is below 7 is acid, and everything above that is termed alkaline, or alkali, or base, A-L-K-A-L. And there's a balance between hydrogen ions and what's called hydroxyl ions. That's what this is all based upon. So as we go down the scale, which is where we're working with our products and in agriculture, 6, 5, 4, 3, 2, 1, 0. Okay, 1, 2, 3, 5, 6. Okay, so on our containers, we always put 5 or below, and there's a specific reason for that. But let's go to the pH scale again. On the pH scale, every shift in number, like from 6 to 7, is a 10-fold increase in hydrogen ion concentration. So, for example, a pH 4 solution would have 10 times more hydrogen ions or more acidity than a pH 5 solution. And looking at it the other way, a 6 solution would have 10 times less than a 5. So it works along that line. Real quickly, and we will look at a chitosan molecule, which is our foundational functional raw material. So here's the basic entity here. When chitosan occurs in nature, or as it's supplied to us, it's in this form. And without getting too chemical, this group here is known as an amine. And in this particular state, in the form in which the chitosan comes to us, this structure here, or this group here, has a slight negative charge. When we make our product, or whenever we're combining this to get this in solution, we're adding acids. And there's a whole range of these that can be used. Now remember, acids give off H+. And so there is a charge attraction here that takes place, and what we wind up with is an H+ stuck on there, and this now becomes positively charged. And that's basically what makes your chitosan soluble. And so in most agricultural applications, a pH 5 is usually pretty good. We supply our products around a pH 2 mark, mainly for product stability. But that's essentially pH in a nutshell. The other thing that we want to talk about with pH is, why don't we want to go anywhere above the 6 mark? What happens at that point is that you're taking away a lot of these hydrogen ions, and so you're weakening the positive charge nature of that amine. And what happens is the polymer chain starts to come out of solution. And we have a small demonstration, a visual demonstration here of 2 different fluids. This golden one here is chitosan in an acid solution. It's clear, transparent. And in an alkaline situation, that white fluffy stuff in here is all chitosan. And for a grower, a distributor, or a farmer, we certainly don't want that in any of your spray tanks or in your spray lines, because it will clog things up. So we want to avoid that, and the way we do that is to give everybody an advisory that keep your solutions at pH 5 or below. Okay, another question that we often get is, why do we recommend the use of citric acid to buffer our tank water before we add our products? And it's fairly straightforward again as to why this happens. So we'll go into our structure of chitosan. We got that. And here is our amine group, which is where all the pH behavior comes from. When we're adding citric acid is a 6-carbon structure, and I'm not going to bother drawing it out, but we'll just call it citrate plus. It has an H+ hydrogen ion component to it, and it's very soluble. It's soluble just as much as sugar is. With our citric acid, what we have here, it's an ion in solution, and we have the hydrogen ion component, which is the acid part, and then we have the conjugate base, the citrate, which is negatively charged. And chitosan on its own is actually another conjugate base. And when we combine citric acid with the chitosan, the hydrogen ion is attracted to that amine group because it's automatically got a negative charge. And the citrate conjugate ion is attracted to this overall thing. So what we're essentially having here is a sort of an acid-base reaction which creates, in chemistry, a salt. So you have chitosan citrate as the corresponding salt that you're generating when you're adding the citric acid to your tank mix. So the citric ion, the citrate ion, actually can carry three available H pluses per molecule. And when we're adding this chitosan to our mix tanks, we're doing two basic things. One, citrate itself is a very good chelator. So the mineral content that's in either the hard water or other substances that happen to be in there can be chelated by the citric acid, as well as the citric acid complexing with the chitosan, forming the chitosan citrate salt. And this is all part of the sort of basic chemistry that's going on inside of your mix tank as you're mixing in and pre-treating the tank mix with citric acid and then adding our material afterwards. So we've sort of gotten all the other components behaving by being chelated, and then we put in our materials and we have a nice stable mix for you guys to go out and spray. How does adding chitosan to the soil influence soil microbial populations and cause various effects in the soil that are beneficial to the plant and the soil microbiota? So what we're going to do here, first of all, is just look at what chitin and chitosan looks like, and I'm not going to draw anything out in any detail, but if we sort of have an analogy of a long linear polymer equating to something like a necklace. So we have a bunch of little structures in here that all repeat. They're all in a linear form like this. And this can go on for thousands of these things. So each one of these dots or these circles is a substituent of the overall polymer. So if we have chitin or chitosan, really doesn't matter. And what happens is when this gets in the soil, these materials here are actually feedstuffs for microbiology, microorganisms. And the way microorganisms feed themselves, they don't have moving mouth parts. They can sense the presence of these substances in the soil, and they respond by producing various digestive enzymes that will break these large materials down so that they could then internalize them into the cell structure to build things of their own. So it's like a foodstuff. So the enzymes that we're talking about here are called, there's two basic classes. We've got chitinases and we have chitosanases. And they're basically the same. I mean, chitosanases generally tend to favor chitosan, chitinases generally tend to favor chitin. Though chitinases can digest chitin, chitosanases can digest chitin, and vice versa. So there's two different types based on their activity. We have endochitinase or chitosanase, and we have exo, the two major classes. And within these classes, there's a whole bunch of different groupings, which we don't need to get into. So what do we mean by these two things? Well, as far as an enzyme is concerned, which is a protein molecule, they can recognize the precise structure of what's within this molecule, these polymers. And if it's an endochitinase, what they'll do is they'll attach somewhere internally along the chain and break the chain that's joining the substituent molecules, and so you wind up with smaller fragments. That's how the endos work. The exochitinases or chitosanases work from either end. So they start off at one end and work inwards. And kind of the way I like to look at it is sort of a Pac-Man type of deal. So these guys just come along and they just start chopping one way or the other and releasing these smaller molecules, which now become water soluble because the polymer isn't water soluble. It's also too large to transport into the cells, the microbial cells. So as these things become smaller fragments, they become more mobile and more soluble and more easy to internalize inside these the microbes. And so what you wind up having is an elevated soil population of these classes of enzymes, which then go on to affect other things like certain pathogenic fungi, pathogenic bacteria, nematodes, all which have or feature chitin or chitin analogs in their structures somewhere. We often hear that people are putting out ground shell that's in a powder or slightly sort of a particulate form. We often hear about micronized chitin, and then we have our stuff, okay? So what's the difference? Well, first of all, this one has chitin, this one is chitin, and this one is chitosan. So there's one immediate difference. The next thing is, if you look at shell, if you look at this sort of stuff under a microscope, just looking at the chitin component, the easiest way to sort of represent this would be to sort of like look at it as a sort of massive squiggle of all sorts of intertwined fibers and whatever else. And embedded in that matrix are a whole bunch of proteins that are either embedded or attached to that chitosan. And then in amongst all of that, again, you have a lot of mineral, which makes these shellfish shells hard. So when you put that on the ground or incorporate that into the ground, microbes now have to get into this and digest it. And the thing to remember is nature neither has a timeline or a profit motive. So it doesn't matter how long, as far as nature's concerned, how long it takes to digest this. It'll do it over time. And it's really the same thing with micronized chitin. All we've done with micronized chitin is we've removed the protein to an extent and mineral. But you still have this fibrous mass that you're putting in there. And even though visibly you can't really see the size of the particle, from a microbe, you're looking at probably half a city block compared to a microbe in a human, for example. So it's a huge structure that it can get involved with. The thing is that this is not that easy to, for the microbes to digest all at once. And so what we wind up seeing, if you were to sort of like graph out how this sort of stuff works, it's not to say that it doesn't work, but it just works so much differently than our materials. If you were to graph out, say, activity and time, with these two materials, you're going to wind up having a very, very slow and very, very gradual sort of activity in the soil. With our material, I can't spell. We have immediate bioavailability. That's the key. And why is that? Well, if you look at it on the graph, you get a much, much steeper rise, which is the immediate bioavailability, and because it's soluble, you get a much higher elevated activity. So why do we have this, or how does that work? Well, what we do, our technology, is we're able to take this material. So our material as it arrives to us is in this form. And what we're able to do is to molecularly disentangle that so that we no longer have this convoluted mass of fibers that are all stuck together that nobody can really deal with. And what we have are individual, clean, molecular strands that are soluble. And that's how you get this immediate bioavailability. And so this would be the chitosan. And we also couple that with, in a lot of our products, yucca. And the yucca is a surfactant. And so the yucca facilitates the entry of these chitosan molecules into the leaf surface and inside the tissues or in the soil interstices. And the yucca also has a high sugar content, which helps to feed some of these soil microbes. High carbon content is always good. And the sugar content in there is very soluble. So you've got a high solubility component, we've got a surface active penetrating component, and then we have our active ingredient in here that's elevating chitinases and giving us very high immediate bioavailability. Well, another advantage or difference, I guess, is that shell is widely available and it could be ground by just about anybody and put out there. It's cheap, which is one of the reasons why a lot of people are using it or tending to use it. Micronized chitin goes through a little bit of processing, obviously to remove the protein and mineral, but those processes are relatively inexpensive, and so this is going to be more expensive than this as a product, primarily because of the processing and the fact that you have to grind it, because chitin's kind of tough to grind on its own. When you get to us, there is a huge leap in technical knowledge, processing, formulating, etc., that goes into making our products. And our products isn't just chitosan. They feature chitosan as an active ingredient along with other proprietary inerts and other active ingredients. But we don't just sell chitosan in agriculture. We sell chitosan as a featured active ingredient. Our materials are supplied at 2%. And the main reason for that is chitosan as a polymer in solution will cause some element of viscosity. And viscosity is a measurement of resistance to flow. So we don't want to make a product that has so much chitosan in it that it won't flow or it won't mix. So there's a number of products that we've seen out there that claim these higher percentages, and there's just no way you're going to get above maybe 4%. You're going to be lucky. Mixing chitosan at 4%. I mean, you're going to need some powerful equipment and some heavy motors to do that. But then again, how do you pour that into a tank? How do you distribute that, et cetera, et cetera? So these people that are claiming these higher percentages are most likely using something like this that's suspended in solution, because this is not a soluble product, even though they claim that it might be. And quite often people inaccurately or accurately, depending on what their intention is, will confuse chitin with chitosan. They'll also state that chitosan is water soluble. Chitosan is not water soluble. You can put all the chitosan you want in water and it will not solubilize. So it's the same thing with shell. You can put all the shell you want in some water as a fine powder and you can make a slurry out of it. You can put it up to 50% probably, or whatever the higher percentages is. But you're not getting the immediate bioavailability that we have.